And y'all can hear me okay? Is that,
Yep.
I have an air filter on low. Okay, great. Because it's pollen season here.
Oh, good. Grace, just let me know when most of the people are in from the waiting room.
I think we have 1 more minute or so.
Okay. There's some feedback. I think the podium needs.
Yeah, I think that.
Oh, okay.
What, Jess, what is it? We have to turn the podium mic off.
Yeah, if you go to like the, room controls, I think just turning the podium mic off solves the problem.
Oh, I think there was a guest in the room who had it.
Oh, okay. Yeah, that would do it also.
So who was like there and watching on the computer at the same time? Oh, fascinating.
Welcome, everyone. We'll just wait 1 more minute before we get started. I know we have a fascinating talk, and I want to give us plenty of time for Q&A. So Grace, shall I start?
Yeah, I think we're good to go.
Okay, thank you. All right, welcome everybody. We're about midway through the quarter, not quite, and I know it's it's always can be a tough time of the quarter, especially spring quarter, but we're delighted to have you here. Very happy to welcome Professor Callie Burt back to our community. She was associate professor at the University of Washington before leaving us to go back to Georgia and Georgia State University. Let me do a few acknowledgements and then provide a brief introduction about Callie for you. But first of all, an orientation. So I know we're all pretty familiar with Zoom by now, but what we like to ask is that if you go to when you have questions, you can post them at any point through the talk. We'll keep them using the Q&A function at the bottom. If you're using Zoom, at the bottom of if you're online, at the bottom of your screen. For those of you in the room, I'm hoping to count on somebody in the room to help with hand raising, and we'll certainly get you included as well. And so I think Jill is there to help us with who's in the room with hand raising. And second thing to mention is we would like to acknowledge a few important factors associated with the University of Washington and CSDE. First of all, a land acknowledgement. We are located on the Coast Salish territory, the traditional territory of the Duwamish, Suquamish, Tulalip, and Muckleshoot nations and other Native peoples, and are grateful for them and recognize what they have given up for us. CSDE's commissioned logo, which you can see on the Zoom, was commissioned from a Native artist, UW Professor Marvin Oliver. And that logo acknowledges the vital connections to Native peoples and territories. This is just a small acknowledgement. There is much more that one can do for the Native peoples to recompense them for what they've given to us. In addition, CSDE is grateful for support from the National Institute for Child Health and Human Development, the College of Arts and Sciences, the Evans School of Public Policy, and Governance, the School of Public Health, the School of Social Work, the Business School, the Population Health Initiative, the eScience Institute, and the Associate Provost for Research. So forthwith, I'm really pleased to welcome Associate Professor at Georgia State, Callie Burt, who is in the Department of Criminal Justice and Criminology and works at the Center for Research on Interpersonal Violence. Their research is been widely published in sociological and criminology journals. And in her work, she's debated the merits of heritability, discussed gene-environment interplay, and the possibilities of future directions for biopsychosocial scholarship. This research presentation stems from a mentored research science development grant, a KO1 from the NICHD. And in that grant, they are currently studying genomics in order to incorporate gene-environment interplay, especially epigenetic mechanisms for embodiment. So I'm really delighted to welcome Callie, and without further ado, turn the floor over to them, excuse me.
No, she's good.
Okay.
So thanks everyone for coming today. I wanted to say 3 quick points before I get started, and with kind of the important part of the talk. And first is it really should have been called Translating the Science of Sociogenomics and Scrutinizing, because part of the issues I'll be discussing is barriers to engagement, given that this is outside the scope of most social science expertise, and why I decided to study this so I could engage in this work. And as related to that, much of this talk will be almost a didactic discussion of some of these issues that we're not familiar with, including kind of background information on genetic advances in the past 20 years that some of us haven't kept up with. And there are going to be a lot of terms that I've tried to minimize terms, but this is nearly impossible in this area. And so you don't really have to keep up with all the terms. I'm going to cover them so you can understand maybe the nuance and complexity, hopefully, but don't think that you have to, if you're not getting the terms right away, that you're necessarily missing out. And finally, I'll note I was definitely overly ambitious when I wrote the abstract for this talk. When I started earlier this week, I put together a bunch of slides at the front end. And then last night about midnight, I realized you have 106 slides and you're not done. So I've had to pare it down a lot. And some of the things we're discussing, I'm really just giving a quick overview of it that I'm happy to talk about more during Q&A, but there's really so much that I could talk about here. I mean, I could sit here for 5 hours, but at that point I'd be paying you all to stay here and listen to me. And I don't think any of us want to do that. So I did want to thank, as Sarah mentioned, this is supported by KO1 from NICHD, and the receipt of which I am definitely indebted to my colleagues at CSDE and my former colleagues at UW Sociology. And Sarah herself was extraordinarily helpful, patient, and so was everyone else. So thanks to them. So sociogenomics is the topic of this talk. And unlike a lot of areas, it's actually a relatively bounded field. And I define it as, and similar to others, as the field aiming to link differences in complex social outcomes, educational attainment, income, risk behavior, smoking behavior, for example, to variation in measured DNA sequence. And this is distinct from some earlier versions of social science genetic, in part because of its use of cutting edge statistical methodologies, which we'll briefly talk about today, and extraordinarily high dimension genetic data. So each individual today in some of these studies can be measured on 10 million different genetic variants. And some of these studies can have upwards of 1 million individuals. And so these are extraordinarily high dimension data. So this work has really been facilitated by extraordinary technoscientific advances over the past 2 decades, which have transformed the way that genetics is done. And in their really important book, and prominent book, The Genome Factor, Dalton Conley and Jason Flesher called this the Sociogenomics Revolution, and they welcomed us to it, noting the fire hose of genetic data that now exists. And we're now able to measure millions of genetic variants across the genome, which is genome-wide, for each individual relatively efficiently and with automated pipelines for less than $100. And genotyping is the measurement of pre-selected variants across the genome. And these SNP chips can now measure 500,000 to over 2 million for less than $100. And so the the other changes that have really facilitated this so-called revolution are, in addition to this lower cost, higher coverage genotyping, major statistical and computational advances, including ones that would have immediately crashed our computers not that long ago. There have been methodological corrections, including ones that plagued earlier eras, especially the candidate gene era, including attention to statistical power, corrections for multiple testing, and things like that. There's also been creation of large consortia to combine expertise in data sets, in particular, given the need for large data sets to test these high dimension data. And in social science, the Social Science Genetic Association Consortium is perhaps the primary group, and it's the most well-known in conducting some of the big mega GWAS studies of social outcome they're getting a lot of attention and being published in some of the best journals, Nature and Science. And their key studies have been on educational attainment. And there have been four studies that I'm just going to show quickly to just kind of show how they look a little different. And these are known as EA1 through EA4. So the first one was in 2013. It had about 100,000 individuals, and it found 3 independent genome-wide significant variants. And the second one, 3 years later in 2016, now had a sample size of 300,000, and they found 74 independent genome-wide significant variants. In 2018, there was a drastic increase up to 1.1 million individuals, and this was due to a partnership with 23andMe, as well as the UK Biobank data, which is about 500,000 individuals. And then within the past month, there's been EA4, which now has a sample size of 3 million. And these are meta-analyzed studies that altogether have 3 million. And they've now identified about 4,000 independent genome-wide significant variants. And if that doesn't mean anything to you yet, then hopefully it will in half an hour. So in stark contrast to some of these prior iterations of attempts to link genetics to social science, which were really often characterized by controversy and resistance by sociologists, this new era is really characterized by acceptance. Critique is generally scarce and muted. And most commonly, the critiques that do occur come from molecular geneticists or biostatisticians, not social scientists. And of course, there are exceptions. And so this work has been increasingly popular, and it's made its way into the media, and it's gotten a lot of attention. These are snapshots from Wall Street Journal, and then 2 from the New York Times about how your success is shaped by your genes and years of education influenced by your genetic makeup. And basically, many sociogenomicists are arguing that given ubiquitous heritability, that we, the rest of us social scientists and population health researchers, need to incorporate genetics into our research. That failing to do so is basically a weakness in our science that opens us up to sweeping dismissals of our research and critiques that our work is fatally flawed. And a number of articles and pieces over the past several years have really sought to introduce social scientists to this work and encourage them to kind of see the promise of this work and consider incorporating it into their own scholarship. And other scientists, sociogenomicists, a smaller number, have taken an even stronger stance and really emphasized not only the potential, but the necessity of incorporating genetics into social science. And some of you have been argued that social science without genetics is fatally flawed. It wastes time, resources, attention, and effort, supports misguided models of human behavior, and misinforms policies, causing still further damage. And so the idea here is that, if social scientists really want to conduct good science that can ameliorate inequalities and make changes, then we need to add genetics to our research. And so this golden age is really what so-called golden age is poised to flourish as polygenic scores, which we'll talk about are genetic summary scores that can be computed following GWAS and other external samples, are being made available as pre-constructed variables in a number of widely accessible social science data sets, including the AdHealth, HRS, Wisconsin Longitudinal Survey. And for example, the AdHealth has over 70 pre-constructed PGSs that scholars can drop into their models just like any other variable. So no genetic expertise required. But as I noted, there's a relative paucity of critical engagement, and there remain current challenges that are often overlooked or misunderstood. And some of the reasons for this lack of critical engagement have to do with social scientists' inability to engage with some of this work. It's extraordinarily complex and increasingly so. The EA3 study that I quickly showed the slide for had over 200 authors. It had 204 pages of supplementary materials, 30 huge supplementary slides, tables, and so it's just a ton of information that necessitates expertise from a variety of areas that one single person can't really be able to follow it all. And then when these tools are being utilized in social science, we're already cramped for space, or at least I always am in our studies, and often some of the key methodological details and challenges aren't addressed. And when they are, these are often really not able to be understood by many of us who don't have expertise in this area. So what I'm hoping to do is begin to kind of address some of this deficit in understanding and help us all understand more what's going on with these studies and what isn't going on, and how we can be better consumers, reviewers, and otherwise evaluators of the scholarship. So hopefully today we're going to, I'm hoping to get through all this to address the following question. So what is being measured in these studies? So do GWASs and PGSs measure genes? Do they measure causal variants? Do they illuminate biological dysfunctions? Do GWASs identify genetic versus environmental influences? And importantly, do polygenic scores or PGSs indicate genetic influences on or genetic propensities for complex social traits as they're often being used? And so, though I'm already a ways in, I'm going to give a brief outline. So I'm going to give a little primer on genomics. Little is relative. An overview of these methods. It's a lot more abbreviated than I would like. Challenges and limitations, and then the main takeaway. And hopefully we can talk more in the Q&A. So as many of us remember, some of us don't, our nuclear DNA is spread on our 46 chromosomes, and this is the focus of human genetics. So mitochondrial DNA aren't generally examined. And each chromosome consists of a linear sequence of nucleotides, which have a sugar phosphate backbone, and the key part, one of 4 nucleic acid bases, so A, C, T, G. And the order of these bases is our genetic code. And we contain about 3 billion base pairs, though this varies across individuals, and females have more than males because the Y chromosome is very tiny. So genes are the sequences of DNA that are scattered across these chromosomes and serve as templates for making a functional RNA product. And most genes that we talk about refer to protein coding genes, and that's what I'll refer to here today. That's the focus. So an example is the BRCA1 gene, which is famous, and it's the DNA sequence of roughly 126,000 bases encoding the breast cancer type 1 susceptibility protein. And so I wrote this, and then I realized we don't have an interaction capacity, but you can just think of it. So how many protein coding genes do humans have in our 3 billion base pair genome? And there could be a lot of answers, but the right one is roughly 20,000. And again, I did this one too before forgetting we can't really engage with each other. Which organism has the most genes? Of course, hopefully you're thinking, if we should put this up here, if humans were the answer, and you're correct, it's not humans. It's in fact rice. So we have about 20,000 genes, and rice has more than twice that. We're about the same as the nematode. So hopefully you can see that organism complexity is not a function of either genome size or genome number. So basically our complexity is in large part due to gene regulation. And only about 1.2% of our genome is protein coding, and the rest was once thought to be junk, but now it's understood to play really important roles, much of it, in these regulatory functions. Although what extent our genome is functional, between 5 and 80%, remains pretty hotly debated. So there remains stuff we really do not know. And so this is intended to show you that our DNA are informational storage molecules, and RNA are really kind of the action molecules. And genes contain ingredients for these other molecules to build proteins. And there are a number of places where these can act and interact in ways that build things that are more complex than what's just encoded on the basically DNA recipe. So human genetics and sociogenomics is concerned a lot with genetic variation. And so we're more than 99.85% genetically similar to each other if we're not relatives. So comparing any two random humans, we would find about 3.5 to 4.5 million differences between them, or about one variant every 800 bases. So there are three key types of genetic variation. And the most common, which constitutes about 87% of the variation between humans, are single nucleotide variants, or SNVs. And these are simply point mutations, an A to a C, and that's what they are. Now, single nucleotide polymorphisms are the variants that are examined in GWASs, and these are a specific subtype of SNVs. So SNPs are SNVs that are present in at least 1%, or some say 0.5% of the population. So SNPs are common SNVs, and SNPs are ancient SNVs. They've become common because they've persisted for a very long time. Most predate the out of Africa dispersal of humans some 50 to 70,000 years ago. New in genetics is very relative. A new variant is one generally that's within 100 generations, or roughly 2,500 years. So new is not really so new. So indels are involve more than one base change, and they're basically any insertion or deletion up to 50 base pairs. And these account for about 13% of our variants. And structural variants are large insertions or deletions, translocations, that account for more than 50 or 100 in some cases base pairs. As a frustrating thing is for me, when I began to learn this, is that like acronyms can have different meanings, they're different standards, and so it's defined different ways. But generally, structural variants now by these large genome projects are defined as greater than 50 base pairs. So most identified SNPs are, well, no, I'm actually going to, yeah, I'll go through this one. So most identified SNVs are uncommon. So there's roughly 500 validated SNVs. Most SNVs in an individual genome are common. That means that most of the variants in our genome are SNPs. They're common. So there's a bit of a paradox there, that most are rare, but most within us are common. On average, there are 30 to 80 new mutations that arise at conception, or actually during meiosis. And so each of us carry 30 to 80 that are not present in either of our parents. And so the explosion of the human population over the past 1,000 and especially 300 years has led to a great increase in these rare variants. And this is important because, as we'll discuss, these aren't measured or proxied well in GWASs, and these are disproportionately likely to be deleterious. So Fu and colleagues estimated that about 86% of deleterious variants are rare new variants. So that's a notable limitation. So most of our variants actually have no known or expected function. So most of our variants that we carry in our genome don't do anything. They're just there. Functional variants are those that affect either the protein product or the amount of product produced, and that should be effect, not affect. So no, that should be affect, whatever. So those that affect stop it, Kelly, or the amount of product produced, gene dosage. So either it's changing the protein or the amount of protein that you're producing. So each of us has about 11,000 functional variants in our genome, including more than 200 that actually make that specific gene non-functional. And that may seem like a lot, and it is in some sense, but in the other sense, that's relatively rare. And recall, we have 2 copies of each gene, so that compensates for some of these non-functional genes. And we have a variety of cellular mechanisms that facilitate robustness to these variations. That said, we see differences in skin pigmentation, eye color, hair color, in part because of some of these non-functional proteins. So to give a quick example of variation, here's a segment of the BRCA1 gene, and you can see here, this is a triallelic SNV, and so it could either be C, G, or T in an individual that we observe. And so C is the wild type that is the functional protein. And if you have a G or a T, it changes the amino acid, and thus it impairs the function of the protein and the action of the BRCA1, the intended function of BRCA1 in terms of its repair protein functions. And so the analogy would be, in a sentence, changing I have a cat to I have a rat. That could be very different despite the one letter, but sometimes that could still work. Here's an indel, and this shifts the frame. And so as you can see kind of in the language example, it really can shift everything that follows this variant. And so these can lead to major impairments in function by just wholly changing everything that follows on that specific gene. So we're almost done with some of the key terms, but a few more. The genome is the DNA in an organism or cell. An allele is a particular variant. A locus is a particular position on the genome, and these can vary from a single base to, you can speak of a locus that is 100,000 or 1,000,000 base pairs. And then genotype is the alleles one has at a given position on the genome. So AA, AC, or CC would be someone's genotype. So why are we talking about this? Well, this is kind of what the genetic sociogenomicists are building on in their work, looking at the effects of genetic differences on behavioral differences. And so the original aim of GWASs was really to understand the underlying molecular basis of trait variation, specifically, diseases, by tracing causal pathways from genetic variants to these outcomes, so we could illuminate molecular pathologies. So GWASs estimate variant substitution effects, like many regressions, if not most. This is the counterfactual change in an individual outcome that would occur from changing the individual's genotype for a particular SNP at conception, holding all else constant. Now, as we'll discuss, variant substitution effects aren't always adequate at examining causes due to things like downward causation and the like. But this is the model that's assumed and that underlies these methods. So what genetic differences are measured in GWASs? So first, not genes or causal variants. Instead, a subset of pre-selected SNPs that have two alleles, so biallelic SNPs. And these SNPs are chosen not because they do something, but rather to tag a region of variation. Now, this section was one that I vacillated about including, but it didn't make sense to say how they tag a region of variation without telling you how that works. So our GWAS methodology is rooted in the block-like structure of our genome. So we inherit full chromosomes from our parents, but these are composed of unique blends of our parents' matching chromosomes. So the two matching chromosomes they inherited from our grandparents, an average of 1.5 blocks per chromosome are blended in meiosis. So it looks like this if you look at the picture on the left, and then we get the blends by the chromosomes crossing over, and you can see with the green and red, hopefully that's not colorblind unfriendly, in the middle. So that's crossing over. And so over generations, so basically crossing over doesn't occur randomly. This is very helpfully. And over generations, blocks of chromosomal segments are passed down unbroken across generations, and these are known as haplotype blocks. So these contain correlated SNPs because, for thousands of years, these have been unbroken by recombination or very rarely broken. And so the association or non-independence of SNPs that exist on these block-like regions or haplotypes is called linkage disequilibrium. And so hopefully I didn't oversimplify that too much. So we pass down blocks. The SNPs are correlated on the blocks. They've been passed down over many generations, such that if we know you have an A at SNP 1, if you're in high LD with this other SNP on this haplotype block, then you're much more likely to have a T than a G at SNP 2. And so we can use that information to both impute genotypes and to tag regions. And so here's a quick picture that my wife said was not informative, but stubbornly I left it in there. And so what you can see here is basically these are hot spots that are you see the white valleys are the hot spots where most crossing over occurs. And the dark red is the degree of LD. And so we can see these blocks have correlated SNPs. So GWASs rely on LD and haplotypes. So rather than measure, any questions, sorry? Oh, so rather than measuring all 6 billion base pairs, which would be impracticable for a number of reasons, we select a subset of tagged SNPs to tag these common regions of variation, to identify regions of association. So the goal of GWASs is not to identify causal variants, but to use these tagged SNPs to tag a region at which the causal variant is presumed to lie. And so we should note that rare SNPs, which I mentioned, are not well tagged, are more likely to be deleterious or not well tagged by common SNPs. Even so, these tagged SNPs capture something because, as we'll see, there are a lot of significant ones. Other variant forms, including indels and structural variants, are also not well tagged by these SNPs. So the goal of GWASs is to use this tagged SNP methodology to capture common variation, tagging other common variation that's presumed to include the causal variant where we think something's happening that's driving trait variation or disease traits. So in GWASs, we have a phenotype and outcome, years of educational attainment, and this is regressed like a normal regression with adding a ton of sophisticated statistical refinements, but at heart, it's a basic regression on each of the millions of genetic variants. So we examine each SNP separately, net of a few controls, usually age, sex, and 10 to 20 genetic ancestry PCs, which capture basically really crude population or really higher, the basic population stratification, which we'll discuss in a second. So these are theory-free analytic approach to scan the genome, without directional hypotheses to see if any of these tag SNPs tag something important. And to do that, these SNP genotypes, AA, AC, or CC, are translated to numeric allele dosages. So you have either 0, 1, or 2 of the risk alleles, which are then treated as a continuous measure in these regressions. So there's several pre and post processing steps that we're not going to examine because the goal here is not to have you be able to do these studies, which takes a lot more time, but there's a lot of pre-processing and stuff that goes on that I'm going to skip over. But one post-processing thing that's really important is identifying lead SNPs and risk loci, because that's what we talked about the 1,271 independent genome-wide associations. And that's how we get, that is basically this slide. So we have to account for LD. So as you'll see here, the figure on the right is a Manhattan plot, and it has these peaks, which are, like the skyline of Manhattan. And these represent correlated SNPs in LD that are tagging the same region. And so they have to account for this, so they're not just measuring things the same effect over and over. And so this is done by basically clumping and picking the variant with the lowest p-value to represent this clump and tag this region. And so risk loci, or QTLs, those are quantitative trait loci, are defined as trait-associated regions marked by approximately independent lead SNPs. And so that's what we were talking about if we refer to this paper earlier, the 1,271, that these tagged 1,271, had lead SNPs tagging independent regions on, approximately independent regions on the genome. And as was noted in the supplement, we think, how much is 1,271? A randomly selected SNP in this study had a 17% chance of being in a risk locus. So, part of the issue with these increasingly large studies is that larger swaths of the genome are being identified. And at some point, if you identify everything, then I'm not sure you've pinpointed anything. They haven't gotten to that point yet, but 17% chance of being in a significant risk locus is not small. What is small is generally their effect sizes. And so I started highlighting that row just so you can see the effect size. And most are less than 0.02 standard deviation units. So most of us, a marginal effect by an increase in having 0 versus 1 allele, 1 versus 2 of the risk alleles. So these are very, very small. And so we can't really use these SNPs and their effect size to tell us much at all about a trait. And biological annotation is very difficult for this reason. It'd really be hard to begin to reverse engineer minuscule effect sizes. And reverse engineering effect sizes would be difficult even if they weren't so small. So just to kind of recover this, and this is probably a slide I should have taken out. The lead SNPs tags risk loci, which the causal variant is presumed to lie. These QTLs can range in size from 50,000 base pairs to over 1 million base pairs. And they often stretch across multiple haplotypes in regions that contain thousands of SNVs, along with structural variants and indels. And there's often multiple genes. There can be anywhere from 1 to, in the educational attainment study, and there's some that had 20 genes in there. And so determining which gene may or may not be the one that may or may not contain the causal variant is exceptionally difficult. And this is why, these studies don't really identify genes and aren't particularly useful for biological interpretation. So while the original aim of GWAS was to use these SNPs to tag regions, to follow up with fine mapping and begin to understand kind of molecular pathologies associated with traits and diseases. In reality, this is proven extraordinarily difficult. GWASs just are not well suited for reverse engineering and understanding the biological pathways to traits and diseases in general, though there have been some successes, but this is particularly true for complex social traits of interest to social scientists. As a result, basically sociogenomicists have shifted from using this for biological interpretation to polygenic prediction. And this has been through the use of polygenic scores. So polygenic scores, and again, sorry everyone for all the terminology and acronyms, are basically genetic summary scores that are created by summing the GWAS-weighted risk allele dosages. So we think back each of those 10 million variants or 6 million variants, we weight them by those tiny effect sizes that we just saw, and then sum them, usually with several sophisticated statistical adjustments to adjust for LD, winner's curse, and things like that. And these are PGSs are used as an index of a continuous underlying additive genetic liability for a trait. So, and we can apply, we can do a GWAS and then use that information to create a polygenic score for each individual and another sample that's used to indicate their genetic liability. And it's been defined as a single quantitative measure of genetic predisposition. So the basic PGS construction process is not that basic. And, but just so you get an idea, you run the GWAS, you replicate the results, hopefully, you adjust for LD, you select SNPs, you then adjust for LD and winner's curse, construct PGSs, test in a target sample, and assess. You don't need to remember any of that for this purposes, really. But what I do want to focus on is the select SNPs phase. So many of us think that PGSs, or I did, think that PGSs were created with the SNPs that were significant in the GWAS. So we pick out the ones that were statistically significant, and then we sum those together and weight them by their effect sizes. But most PGSs are actually created with all SNPs, not the significant ones. And this is because the process usually consists of creating PGSs at a variety of p-value thresholds. So they use 5 times 10 to the minus 8, 10 to the minus 5, 10 to the minus 3, or p is less than 1, which means no p-value threshold, and then seeing which one predicts the outcome best on the basis of R squared. And this is almost always, but not always, but usually the one with no p-value threshold. And so if it predicts the outcome best, it's selected as the PGS for use moving forward. So, most of us didn't realize that the statistical significance isn't really relevant. Then these are used to create the all SNP PGS. And these are found to be much more environmentally confounded than the ones with lower or more stringent p-value thresholds. And that's because even subtle biases that we're going to discuss in a second can really accumulate as these millions and millions of SNPs are being added together. So what are PGSs useful for? So sociogenomicists have argued that their perhaps greatest value is as a control for genetic heterogeneity. And so the idea here is that we can really get genetics out of the way by incorporating a PGS so we can more clearly see the effects of the environment. So that's one of the main ones. We can show, hey, education does matter even when we control for genetic predisposition. As a second use is as a powerful analytic tool for causal influence, and scholars have pointed to the potential of PGSs as a molecular tracer that they can use to observe the flow of students through educational systems. Another use is gene-environment interplay. And the common analogy here is that PGSs can be used as a prism for G by E models to see how treatment effects have a rainbow of different effects on different individuals as a result of their different genetic propensities or backgrounds. Another use is risk stratification and early identification, particularly the so-called at-risk individuals from the upper and lower deciles of PGS scores. And finally, scholars have argued that PGSs can be used, and just genetics in general, to really shift our worldview and understanding of social inequalities. And so on this view, PGSs reveal the hitherto unrecognized fact that success in life is partly shaped by our genetic inheritances. And so these are the main arguments for the use. It's not fully comprehensive, but in my short remaining time, I want to basically speak to some of the challenges with PGSs that undermines their utility for some of these purposes. And specifically, we think about PGSs as a control for genetic heterogeneity. And this only really works if genetic environmental influences on traits can be separated into a PGS and an environmental effect, and that PGSs are then relatively accurate and unbiased estimators of genetic influences. And as we can see, particularly for complex social traits, this is not the case. So GWASs cannot really disentangle genetic from environmental influences, such that PGSs do not capture genetic influences versus environmental ones on complex social traits. And so the challenges and limitations that I'm going to now unfortunately have to quickly move through are environmental confounding, low resolution biology, and then context specificity. So I could really talk about an hour on population structure and phenotype stratification, but instead I'm going to give myself 2 minutes. So basically population structure, and this is one of the main challenges of genetic association studies. This refers to systematic differences in allele frequencies between subpopulations due to different demographic histories, migration, admixture, which often appears as geographical structure due to ancestry. And because subgroup membership is often associated with trait associated environmental influences, population structure can bias genetic associations. So we end up with uncontrolled environmental influences being correlated with subgroup membership in a manner that creates spurious or inflated genetic associations. And the classic example of this is a GWAS of chopstick use. And so this is from Hammer, who came up with this, building on some others. So if we were conducted GWAS of using chopsticks and a sample of diverse ancestry, while we might find some significant associations related to finger dexterity, most of these associations would be due to cultural differences, namely random variants that differed in frequency between East Asia and the rest of the world, and had nothing whatsoever to do with a genetic propensity for chopstick skills. So in practical applications, phenotype stratification is most plainly manifest, as I said, in this geographic patterning of polygenic scores, which really reflects sociocultural and physical environmental influences. And we can see this in one of the more famous studies here that should say genotypes, but we can use the genetic ancestry. We can basically use these SNP matrices to create PCs. And this is the PC1 and PC2, which I should note are 1 through 10 are usually controlled in most of these studies, but these map onto geography. You can see here slightly rotated, it really resembles Europe. And this is a famous picture. And they also use this to estimate location, which it does work quite well. And this is the basis for, you send a 23andMe your spit, and then they give you information about your ancestry. And this is related to the different mostly random alleles that you possess that can be used to kind of correlate with geography and thus ancestry. So there have been several sophisticated methods to reduce or mitigate population structure confounding. I mean, this is a known issue that's been dealt with for many years, and genetic PCs, genomic control, linear mixed models, and LDSC. And I can't go over those. What I can say is these do appear to reduce population structure confounding, but they do not eliminate it. And this is particularly true for complex social traits. So uncontrolled population structure persists in these studies. And it's exacerbated under the very modeling conditions most often used in social science, sociogenomic studies. That's when their studies, including small ones, are meta-analyzed, and then these small effects are aggregated into PGSs, where even subtle biases can substantially add up and influence the PGSs. Familial confounding is another concern. We not only inherit, 1/2 of our genome from our parents, or our whole genome from both of them, but also their environments, social status, culture, et cetera, social networks, and which is of interest to most sociologists and social scientists. So the association between parental genotype and offspring genotype and then trait are confounded by the association between parental genotype and parental environment. And these effects may be amplified over generations through processes that are called dynastic effects, as disadvantages and advantages accumulate, as we well know. So several innovative studies have illuminated the extent of familial confounding in GWASs and PGSs, and these indicate that as much as half of the observed effects in these studies are due to familial confounding. And they can do things like examine the non-transmitted parental alleles, adoption studies, or just do straightforward things like controlling for parental education in models. A third issue, and one that what I'm particularly interested in, is downward causation and artificial genetic signals. And so downward causation is defined by me as social cultural forces that select individuals based on different traits, which are to some degree genetically influenced, into different environments and exposures. And these create artificial genetic associations. So these are environmental influences masquerading as genetic influences in GWASs, because GWAS's counterfactual model cannot distinguish between downward and upward causation. And so the classic example of this is Jinx's red-haired discrimination example. And so he asks us to imagine a system where red-haired children are barred from school. So in such a system, if we were to do a GWAS of educational attainment, genetic variants linked to red hair would be identified by GWASs as causes of lower educational attainment. However, most of us would recognize that neither an individual's red hair nor the genetic variants contributing to red hair are appropriately conceived of as genetic causes of lower educational attainment, right? The difference that makes a difference here is not red hair, but the social structural norms and institutional constraints that bar people with red hair from schooling. So, of course, explicit discriminatory policies like these are thankfully no longer present in much of the world. We certainly have ongoing and persisting discrimination, as well as the legacy of past discrimination, which can shape downward causation and artificial genetic signals in contemporary GWASs and associations. And so this is an important factor to consider, especially when people are arguing that GWASs and PGSs reflect genetic potential for achievement. And the final weakness that I cover here is low resolution, basically, or limited coverage of genetic variation. And so, as I talked about, GWASs use tag SNPs to tag regions of common variation, but they do not tag rare SNPs well. And there's limited and uncertain coverage of indels and structural variants, which research suggests are more likely to actually be trait relevant than even SNPs. So that limitation is significant when we're thinking PGSs can represent genetic differences. They are proxy for some genetic differences is what they are essentially that matter. So they also focus on additive effects, and we know from cellular biology that protein-protein interactions are the norm rather than the exception. So the idea that we can just sum these things up and get prediction of liability is necessarily oversimplified. And that's the main story there. So I lied, we have one more limitation and then I'll quit talking. So the final limitation is context and population sensitivity. And basically, like heritability studies, GWASs and PGSs tell us what are the associations in a current context, not what can be changed, or what is elsewhere. And this exists for both biological reasons and basically sociocultural reasons, right? So when we think about traits that facilitate, social behaviors and achievements, they vary depending on context and relative conditions. So the example I use is the traits facilitating educational attainment and achievement for a woman in the 1890s USA, 2010 Saudi Arabia, 1950 Nigeria, and 2022 USA are likely to be distinct in non-trivial ways. So a woman achieving going to higher ed in 1890s USA would be deviant, whereas that same person in 2022 USA would be conforming. So the idea that we can then use these genetic associations across context and condition as a static genetic influence is not quite right. So finally, I'll conclude basically with this rushed talk about the implications of these. So why does this matter? This matters because PGSs and GWASs are often characterized as representing genetic influences on, or even more concerningly, propensities for these various outcomes, educational achievement. So we can, some studies speak of the high genetic potential students leaking out of the STEM pipeline. But these do not reflect genetic causes or influences or propensities, but correlations in a given context that don't account for downward causation and environmental confounding, including the legacy and of discrimination and wealth and class advantages over time. And so PGS is they're low resolution. They're not measuring causal variants or genes. So a recent study talked about mothers with more, quote, education-related genes, which that doesn't even make sense. And that's not what we're measuring. And finally, PGSs can't be compared across context and condition. So since I've talked so much, I'll stop there and leave some time for questions. And I'm happy to elaborate on anything because I know I did a bit too much, but I did it anyway.
Thank you, Callie. I don't see questions. Here, I have Dan Eisenberg. Let me see. I'm going to ask Dan Eisenberg to join. I think you probably know Dan. Dan, you can talk.
Can you hear me okay?
Yes.
Yes, thanks.
Good. Thank you for the talk. Good to see this progressing, and wish we had more time to delve more deeply. I wanted to ask a little bit more about the idea of statistical interactions and the non-additive effects, and this idea that protein interactions imply interaction effects. And I'm not fully sure of this. And it's an idea I've discussed with other bioanthropologists and others, that two proteins interacting with each other, and even many more, could imply statistical interactions. But really, when we say interactions there, I think we mean something different, which could overlap. So and maybe you're thinking of protein interactions which would imply statistical interactions, but it's not necessarily clear that that molecular biology definition of interaction is the same as what we mean as statistical interaction.
Yeah, great question and nice to chat. I mean, one, I think that was I normally don't think I even would have said that specific statement, except, I was like, how can I say this as quickly as possible? So I honestly think you're right in terms of it's not necessarily true that protein interactions at the cellular level translate to statistical interactions. But I think that's a short way of also recognizing that if we have a genetic variant affecting something, that it's going to potentially interact with other variants that we have. I mean, I think that we do know that, and that it will compensate for and interact with and thus shape traits that, so we're less interested in the protein it produces, right? And we're more interested in its phenotypic manifestations, which I think we know, given the way organisms work, that necessarily is interactional. Whether that is detectable in statistical interactions I don't think your question is. I think you're saying whether it's important. I think we're not sure. And so maybe you would speak to kind of the studies that have looked at, at using genome-wide, variants, looking for variants QTLs and to see if they exist, and they don't really find a lot of evidence for it. But I would sort of argue that, using these low resolution methods to try to determine, to basically decide whether or not we think they exist is really not the right approach. I think we have to know that we understand they do exist, and then potentially think about how that affects what we can know and cannot know from these methods. Do you agree?
Yeah, no, that makes sense. And I like that you're pushing it to what we actually know there. And I guess the other area that arises, and feel free to kind of not delve too deeply into this because we don't want to lose the rest of the audience, is my understanding is the evidence from heritability studies is kind of suggestive of more additive effects being the kind of primary factor, but I don't know how reliable that is. And again, feel free to kind of defer if that's going to get too in the weeds here.
Yeah, I mean, I would think, I have a lot of issues with heritability studies. And I think almost necessarily the assumptions that they make lead to the finding of more additive influences. But I mean, I think that these are really interesting questions that we're not really able to answer right now. And so I think, we can say, we know this exists. And so we need to think about how this may be limiting what we can find. But I think you're right in terms of the extent to which it should influence the statistical findings and the substantive findings is an open question. And So in that sense, I may agree.
Thanks. I'll let others jump in.
Thanks for your questions.
Thanks, Dan and Callie. Steve Gaudreau has his hand raised.
Yeah, hi. So thank you, Callie. That was really fascinating. And I'm also a biological anthropologist. I imagine this is not a coincidence. And my question's a bigger picture about the politics and everything here in terms of, having been trained all the way through in bioanthro and been in the field for 25 years, I've sort of seen the previous waves of, studies of genetics and on behavior and social life, and then the critiques of those, and lots of people talking past each other and getting very mad at each other and assigning political motives to each other and then giving up and and all that. It's great to see kind of a new generation of scholars saying, no, we're going to keep trying to do this and be level-headed about it. And your critiques were really, I thought it was very clear and level-headed and and fair and all that. So I guess, but with ending with all the critiques, I guess I'd ask you, what do you see as the big problem? Like given all the critiques you have, and the challenges and the limitations, where do you think this is headed? What do you think is going to come?
That's a great question. I really don't. So I do think that that they're going to become more popular. Much like I think we see the new methods arise. We can do them. We try to see what they can tell us. In terms of what I think their actual utility is, I'm really not sure. I do think that I personally am on the more skeptical side, because I do think there's been an enormous amount of waste in the candidate gene era, right? Like Matthew Keller, psychiatric geneticist, said something like 20 years and millions of dollars for nothing. So I think there's a danger in doing things just these are exciting, statistically significant, sophisticated tools that we can do a lot with. But I want us to kind of think more deeply about the substantive value added of these and kind of think about, okay, what can these be used to do in a manner that doesn't reify PGS as its propensity, and think about that. I mean controls for something, but not for genetic heterogeneity. And so I think there are some uses. I think they're less promising than they're being touted as at present. In part because we have a lot of things to figure out. But I share your. I think it is important that I think the past it was hostile, the debate, and basically one side yelling you're anti-scientist and anti-bio, and the other side yelling you're racist and eugenicist. And I think that's not useful for anybody. So I do think that's important to engage in this with the scientific issues and consider ethical implications and sociopolitical ones, which are obviously important, but recognize we can speak about the science and the implications, and that these need not be confounded.
Super. Thank you so much.
So basically, I'm not sure. That's what I'm still sort of thinking.
Yeah.
Do you have thoughts on that? I'd love to know if you have thoughts on it.
Yeah,
okay.
Well, I don't work in this field, but I largely agree with you. But it'd be great to hear what Dan or others think as well at some point.
Yeah.
We should shut up and let others. I'll be quiet. Yeah. But thank you so much.
Yeah. Thanks.
This is Sarah. I might take the prerogative here. It's great to see where you are with this work. And I feel like I just, not being a biological person at all, it was like a fire hose to absorb everything that you just told us, but it was very informative and helpful to me. I guess where I thought you might be going, which is a direction where people in the big data world, data science, social media, analysis of social media world, but is let's put, rather than rushing to do sort of inductive sets of associations, let's think through a study designs that, so using our social science toolkit of thinking about heterogeneity, thinking about what is our denominator. I mean, all of these really valuable tools that we bring to the table as social scientists, struck me that there are inklings of that in what is now being talked about, but that might be where you're headed. Like, let's be very, very careful in our methodological study design and epistemology and bring those to bear on this in this world in an interdisciplinary way, that maybe there's enough room for that kind of collaboration and appreciation.
Yeah, that's a really great point. If I'd gotten to, I did have some slides on suggestions that were they're down there, and I didn't quite make it to them, and it includes, some of these studies are really like, we have a social outcome, now let's apply these statistical methods to them, and kind of basic social associations that we know exist, social class, geography, are not included as controls. And to me, that's where we have to be like, we know these exist. We can't examine educational attainment genetic associations without controlling for these fundamental structural differences between individuals. And so I do think that's where it's sort of like, there has to be a no. So I have a things of do's and don'ts that I didn't, make it to, but basically it's about, for even if you're reviewing one of these studies, check and see if these kind of basic social, influences are controlled. And if they are, what does that mean? Even things such as, controlling for geographic region and social class, which most of these studies have even kind of rough measures. So, I think that's where sociologists can add, downward causation, and kind of, especially if we're looking at these complex social outcomes and thinking about, how we're even measuring things like educational attainment,
Right.
Years of educational attainment is, it exists, but I think we know that doesn't measure intelligence. Some of the smartest people, never had the chance to go to college. Some of the, and there's so many different things. It's not like there's a unidimensional trait for intelligence.
Yeah.
So I wholeheartedly agree that there's a lot of room to engage with this in ways that doesn't just mean drop the PGS into your study. And by no means am I suggesting that all scholars are suggesting that. I'm just amplifying that argument.
Right. Can you share
great.
that slide of do's and don'ts? Can you pop that up there?
Yeah. So the first one is
Thanks.
speaking to your point, do evaluate whether known socio-environmental controls were included in the study. And that's really a key one. So, even articles that have appeared recently in American Sociological Review, American Journal of Sociology, they haven't controlled for class and looking at kind of an educational attainment PGS. And for that, we've kind of got to be, wait a second, that needs to be controlled. And so I think these are the kind of things that we'll begin to do, hopefully sooner than later. And I really have, strong opinions about the use of the term propensity, which I think is problematic for, a number of reasons. One, that's just not accurate. And I think there's an absolute danger of using propensity and then potentially kind of reifying inequalities by viewing people who are, positioned the way they are due to, accumulated disadvantage, and then calling them, up, well, they couldn't get much because they have low propensity, when really it's nothing about that. And so that too is a major concern. I think that hits all the big ones, except I also didn't mention, these are not useful. I can't believe I overlooked this. These are not useful for individual prediction. So you cannot use a PGS to predict an individual. It's wildly inaccurate. Almost everyone except Robert Plomin acknowledges that, and maybe his colleague Ashbury, but everyone else acknowledges that they're not useful for individual prediction. So that is out.
Yeah. Well, thank you. We're at time. You're welcome. Actually, I don't know if Dan wants to stay on and chat with you, but you're certainly welcome to stay and chat with Dan. I don't know if he looks like he's already taken off, but I'm sure everybody would love to hear from you again. We'd love to have you come back again. Thank you so much for coming.
Yeah, thanks
Callie.
a lot. Thanks everyone for coming. And sorry for having to cover so much. Next time I'll do less genetic specific stuff.